for this, so you guys can look back on it. But okay, so that is, so presentations are a must. I've already assigned everyone a question. Let me know if you guys can actually see them or not. And if you have any questions about those questions themselves. So these presentations are obviously 5 points. A PowerPoint is required. Definitely state the, so the points will be evened out like this. So stating the question will be a point. Giving enough background information to answer the question, and getting the actual answer correct. So those are the 3 main components I'm looking for in a presentation. So I'll give you an example. So this is a question as an example for a presentation. Which nitrogenous base is shown here? So we have the structure, and it's asking which base is it. So seeing this, so now I've stated the question, right? So next we go into some background. So we see this base. We see that it's a double ringed structure. When we see that, you say it's purine, right? It's a purine. So once you see its purines, you can knock out cytosine, thymine, and uracil because there are purines and single structures. Then you have adenine and guanine remaining. So in guanine, the distinguishing factor that we have is the carboxyl group. So when you have that, it is guanine, and you do not see a carboxyl or a C double bond O in this structure, and you see an amino group or an NH2 group. So that cancels out guanine, which leaves you with adenine as the right answer. Does that make sense in how we should be doing presentation questions? Like how, like give the, like you state the question, give the background, and then state, narrow it down, essentially. Does that make sense? Yes, no, maybe?
Yes.
Okay. Okay, so the structure that I'm looking into for now with discussion sections, for one thing I can tell you right now is that discussion sections are supposed to supplement the lecture portion, and it will be focused on major concepts. I cannot go through everything in every little detail that Dr. Osuna covers it as. So I can go over main concepts. If you guys bring any questions, we can cover those too. But the way I want to run this section is like this, but I'll tell you some other ideas that I have as well, and then you guys can choose which one you're comfortable with, okay? So with so this is going to be the synchronous version of discussion. So discussion videos with the ones that I'll be providing for you guys, they focus in on the concepts I will be covering. But these videos are definitely not a substitute for a lecture. So please keep that in mind while you guys watch these. It's just to give you a different perspective on the different major concepts. You have quizzes. The quizzes will be available with the videos on Wednesday evenings. The first set will be up tonight, so you guys will be all set. The password to the quizzes are actually in these videos, so definitely do watch them, because it does take me a lot of time to make all of this happen. Then we come into these sessions, like the discussion sessions themselves. They will be held during this hour. We will be doing presentations, attendance activities, and whatnot. Again, covering anything that we missed. And this is where the attendance and participation points come in, okay? So in a way to make this more, I guess, individualized and help you guys more, I decided to let you guys choose on the different ones. I'll probably go with the one the majority chooses between all three of my sections. But I will, we can talk that out if anything. So synchronous is the version that I described so far. Now with asynchronous, presenters can come in, are the only ones that have to come in. So the assigned week, when you guys do the presentation, that's when you guys should come in. In this case, the attendance points get negated out because not everyone will have to join us, so that will be negated out. Again, if and the quizzes will revert back to 20 points instead of 10. Whereas with self-paced, we won't meet weekly, but the quizzes will still remain weekly, but you will still get credit for them as long as you get it done before the lecture exam, okay? And the presentations in the self-paced one will be self-recorded and emailed to me by the end of the week of the scheduled presentation date. With all three, we can still run what's the word? Like review sessions if needed. Everyone can join in with okay, hold on guys. Okay, hold on. I'm gonna give you guys a form to fill out because this might so you guys can answer on there which one you guys actually want to do. Let me just get that link and I'll send it in the chat so you guys can fill that out for me now. That way I don't lose any of your information. And it's like attendance points for today. Okay, so please take a look at the forms that I just sent you. Let me know if everyone can access it.
Yeah, we can.
Okay. Do you guys have any questions or concerns? Other than the fact that this is very late?
No.
I actually do. Around what time do you post the quiz?
I should put it later tonight, but you can do it anytime during the week.
Hi. So
For the presentation, can we like go to office hours to get help from you if we don't understand our question?
Yes, of course. You guys are welcome to come bug me during my office hours anytime.
Okay, thank you.
Yep.
Excuse me?
Yes.
So I'm looking at the self-paced option, and it definitely looks like something I'd vote for, but the only thing I'm worried about is I don't necessarily know a good way to self-record like a PowerPoint and then send that in. Would you happen to know any way that, like an easy way to make that work? Or
You can, so through Zoom, you can actually record yourself with the PowerPoint in it, like the way I'm sharing it with you right now. If anything, if you're leaning towards self-paced, but you don't want to do the self-recorded version, then you can do asynchronous, where the presenter only has to come on the day that you present. And you can just do it there.
all right, thank you.
Yep. Any other questions, concerns?
Hi, I have a question.
Yes.
On the form, it says, what do you guys want to see out of discussion? Like, what do you mean by that?
So so some of the things I'll be doing is like videos, or like finding different videos that can actually supplement more of the stuff. Sometimes study guides or like detailed versions of them. Whatever you guys like. My job here is to help you guys understand the material more. And the way I can only do that is understand what works for you in studying wise. So like you can say, oh, I'm a visual learner, so I want more videos, or I'm a reading and writing kind of person, so detailed study guides, or something like that. Like whatever, like I guess I'm trying to figure out ways to make discussion more helpful to you guys in understanding and being awesome in genetics. So that's kind of what I meant up with that. Does that answer your question?
Yes, thank you.
Okay.
So for the quizzes,
Mhm.
so you're gonna post the one tonight and it's due next Wednesday, and it's gonna just be the same
Yep.
thing over and over. Okay.
Yep. It's gonna, every time it's gonna be due Wednesday.
Okay, thank you.
Hi, sorry, can you hear me?
Yes.
So no matter what option we choose, you're going to post like pre-recorded lecture, or not lectures, but discussions.
I am gonna have the videos up just because I want to make sure that I have enough, because like I cannot go through everything in the 55 minutes that we have. So I want to make sure that we get through the concepts, review it's a way to allow you guys to review the concepts even more. So I'll have them for all three
Okay.
versions. Even if you guys choose asynchronous or self-paced, I don't know how it will work with self-paced, but with asynchronous, if you guys still want a synchronous version of it, I'll still have other problems going on with within the presenter session, and put that recorded on Blackboard. Any other questions? Are we all on the same page?
I would have a question.
Okay.
Are you going to tell us which one we're going to be using from now on?
It's a bit, I think too much sound is coming in. What did you say?
Are you going to tell us like what we're supposed to?
Okay.
Can you hear me now?
Yes. It's like
Okay, so you're
echoey,
gonna tell
but
us-
yes.
After we're done choosing, are you going to tell us what we're going to be doing like for the next week?
Okay, that,
Would that be asynchronous or synchronous or like self-paced?
Rafi, can you
Maybe
send it through the
chat.
chat, please? Ooh. Rafiq, you okay? Okay, are you going to let us know what we will be going by after we vote? Yes, because that's the only way we will be able to do anything further. So I will let you guys know what all three of my sections actually decide by email. Oh my God, it's a three-way tie. Okay, this is interesting. I'll figure it out at some point. Okay. Wow. Wonderful. Okay. Anyways, I will let you guys know what I decide. It'll probably end up the synchronized version. Oh, wait, no, hold on. Sorry. Oh, no. Come back here. No. Jeez. Okay. You guys erase everything that you just saw from your heads. Okay. I was trying to look at something here, but okay, I think everyone's actually leaning towards synchronous. So I guess we're doing this synchronously.
Yay.
That'll be fun.
It's staying synchronous, right?
Huh?
It's synchronous.
Yep, synchronous.
Yay.
Yeah. I just wanted to make sure you guys had the option. But I guess synchronous it is. Great. Okay. So any other questions about the discussion in general before we move on? We good? Okay. Okay. My little nematode, stay there. Okay. So name the following nitrogenous bases. Which two are these? Anyone?
Guanine and adenosine. Adenine. The A one.
Okay, which one is what?
Is the one with the H
one with
with the A?
Sorry, you go.
Oh no, it's okay. You got it.
No, you got it.
Somebody answer,
Okay, so
please.
the one with the H2N is the one with the A, right?
Yes, that is adenine. So we know these two are double ring structures. That is adenine. And this guy with the carboxyl group, which is the C double bond O, that is guanine. Are we cool with purines? Okay. What about these guys? We know they're pyrimidines. What else do we know?
One ring.
Wait, say that again?
One ring.
Yes, they are single ring structures. So what can you tell me about this guy? The one on the far left.
I believe that is. Geez, I don't get it wrong.
Okay, so when you see an NH2 or an amino group, think cytosine, okay? With
All
these,
right. I didn't want to guess that.
so whenever you see NH2, think cytosine on a single ring structure, okay? Next, these two look very similar, but the difference is this methyl group. What is this guy? What is the one in the middle? Huh? Oh, somebody said it. Thymine?
Yeah.
Yay! That's thymine.
And uracil.
Yep. So when it's this alone, uracil. When it's these two combined with the methyl group, that's thymine. Are we good with identifying these nitrogenous bases? Okay. Next, we go on to this question. If an organism has 10% adenine in their genome, what is the percent makeup of other bases?
Wouldn't it be
45, 40, 40, 40, 10.
Okay.
45, 40, 40,
Why?
40, 10. Yeah. 40, 40, 10.
Yes, the answer is correct, but why? It'll be my favorite question throughout the semester, so why? Why is the answer 40, 40, 10?
Because it's a percentage, and when you add them, you get 100.
Okay, that is definitely correct.
Isn't it like the formula for like where there's
This is like cell bio 1. I mean, oh, my thing is over.
Okay.
The 10 is for the complementary base,
Mhm.
and the 40 is just, well, to me, the right number is just to add up to 100.
Do you remember what rule that is?
the complementary rule?
Yes. It's also called Chargaff's rule for base complementarity. It says that adenine is proportional to thymine, and cytosine is equivalent to guanine. Okay. So knowing that 10% adenine means you have 10% thymine. Then, as you guys said, we look at the percentages, 100 minus the base pair, the given ones, and then you kind of get 40% cytosine, 40% guanine. Does that make sense in explaining why that's how much it makes up everything? Okay, good. I like seeing thumbs up. DNA and RNA are chemically similar, but are distinguished in large part by the 2' hydroxyl group in RNA and 2' hydrogen in DNA. Why do you suppose that we do not typically find nucleic acids within the cells that have 3' hydrogen groups? Do you guys know why?
It
No.
wouldn't bond with the other 3 hydrogen groups that it would need to bind with in order to complete the double helix, or at least the DNA.
Wait, are you talking about the hydrogen bonds that are in between, or the phosphodiester bonds?
Oh no, the hydrogen bonds.
Okay, so the reason why you do not see 3' each. Okay, so we're talking about the sugars here, not the bases. So when we look at the sugar, the 3' hydroxyl group here goes into the next carbon's phosphate group, right? And form that phosphodiester bond to link them up, right? So without the ability to create these bonds, you cannot make the polymer or the chain that is DNA or RNA. Does that make sense?
So basically, it's because they can't make phosphodiester bonds.
Mm-hmm.
But
Does that make sense? No?
Kind of.
Okay, so what is the confusing part at the moment? So the question is asking, why do we not, so you know how ribose, and so this is ribose here, right? With the OH at 2'. In deoxyribose, you don't have this hydroxyl group, but you still have this hydroxyl group. This question is asking, why do we not see a 3' hydrogen instead of this guy, right? That's what the question's asking. The reason is this 3' hydroxyl group is what's required to form a chain of DNA or RNA, because that's the ones that are forming the phosphodiester bonds that make it linked between the sugars and the different bases. Does that make sense? Yay. Okay. So what do you guys know about Griffith's experiment? What did he discover back in 1927?
The virulent and avirulent cells. Is that Griffith?
Yes, that is part of it. Anyone else want to add on?
The transforming principles.
Mm-hmm. That is one of the main components, yes, okay. So Griffith used 2 strains, the smooth strain and the rough strain. So the smooth strain was the virulent one. When he injected those cells into the mouse died. When you had the R strain or the avirulent ones, the mouse ended up living after getting injected by it. Then Griffith heat killed the virulent strain and the mouse lived. So the next thing he decided to do was see if you put the heat-killed S strain and the avirulent R strain together and put it into the mouse. Griffith thought that the mouse was going to live, but as a result of this injection, the mouse actually died. And when they analyzed the tissue, they saw living S strains. And they were like, what? Like, what's going on here? That shouldn't be right. It's like, maybe this something in the S strain is actually transforming this, the avirulent cells. So they were like, okay. That is essentially what Griffith was up to back then. Does Griffith's experiment make sense so far? Okay. So the Griffith experiment showed that certain strains of Streptococcus bacteria could transform from one strain to another when combined. How did Avery, McLeod, and McCarty expand on Frederick's observations? So how did the three scientists expand on Griffith's work?
I think it's A.
A.
Why is it A?
Because when they tried it with the first did it with the protease, and then they realized that protein was not a transforming principle, and then they did it with RNAs, and then they realized it wasn't, and then they did it with the DNAs, and then they realized there was a change, so they realized that was a transforming principle was DNA.
Yep, that is exactly why. So they did these experiments. They saw that since a transformation between these guys didn't occur, they said that it is not protein or RNA. But then when no transformation occurred when treating with DNAs, then they were like, aha, no transformation. So voila, DNA is the active factor that is transforming these cells. Does this experiment make sense to you guys? Okay, great. So that is how the answer is A for this question. Okay, next question. A famous experiment demonstrated that DNA could direct viral replication, providing evidence that DNA is genetic material. Which scientists were directly involved with this experiment? If you guys don't remember, we can narrow it down by process of elimination. So we know.
Wasn't that the same thing we just talked about, like the Avery, McCarty, and Blade? Wasn't it that one?
So with that, it's more okay, so that was not virus-based. That was more bacterial, where they were showing the different different effects of them.
Is it for Griffith?
Wait, say that again?
Griffith?
E?
Oh.
Oh, everybody's talking at the same time.
D.
I was saying B for boy.
I was saying D for dog.
Okay, so Griffith is not viral. That was Streptococcus pneumoniae, which is actually a bacteria.
It's E2 phage bacteria, not virus.
I thought it was D, Hershey and Chase.
Yes.
I thought Hershey and Chase used T2 bacteriophage and not virus.
So bacteriophage is actually a virus that infects bacteria.
Oh,
So that's the key word that involves there. So phage is another fancy word for virus.
gotcha.
Bacterio is bacteria. Bacteriophage is a phage that infects bacteria. Okay. So what is going on here with Hershey and Chase experiment? What is the first thing they do?
They infect the bacteria with the phage.
Okay, the first thing they do is radioactively labeling DNA and protein, right? Then they go into infect, they are labeled, and then they infect the bacteria. After infecting them, they put it in a blender, remove all the phage from the bacteria surface, and then they go on to showing that the phages are unlabeled when it's DNA that is incorporated, and the bacteria gets labeled with the P32. Whereas with the sulfur 35 one, they saw that the proteins ended up labeled in the phage itself and not in the bacteria. So that's how they kind of confirmed that DNA was the transforming factor, or what is being passed down as the genetic material. Does that make sense?
No,
Where did I lose you guys?
the blender.
The blender, okay. So the blender part is just to remove these guys. So the phages are infecting the bacterias in both cases, and they're like, okay, what do we see here? The phage bacteria here are not labeled, which means something was passed through. But we see that in the bacteria here with the DNA labeled, you see that the bacteria was actually labeled, like literally lit up like a Christmas tree. So in this case, they saw that the P32 incorporated DNA was actually transferred into the DNA. But here they saw that something was transferred, but they still, the protein that was radioactively labeled stayed within the phage.
So to clarify, the phage is just like the protein shell, right?
Mm-hmm.
Gotcha.
The phage ghosts are essentially the shells without their genetic material in them. Does that make sense? Are we okay?
So what was like blended that made the difference?
Huh?
Like, you know how like one has the radioactive and one doesn't? Like, what made the difference? Like, what did they blend differently?
Or was it
Okay.
like determined because of the phosphorus? Since DNA has phosphorus and
Mm-hmm.
protein has sulfur.
Yep. So the thing with so the blender part is kind of like a way to remove it out, like letting the separating out the phage and the bacteria. So in this case, since DNA incorporates phosphorus 32, because of the phosphate groups in the DNA backbone, they were seeing that led to so the infection part is to allow the bacteriophage to transfer its genetic material into the cell. So whatever it was, so in this case, since it was the DNA that was incorporated here, you see that the bacteria ends up labeled because DNA is what went through. Does that make sense? It's a similar scenario with the protein part because they're seeing the phage ghosts or the phage shells labeled with the S35, which proved even more that proteins are not the genetic material. I know it's a little confusing, but hopefully that cleared up a bit. Okay, great. One more question and I will let you guys leave. So, how do bacteria package their DNA? I'm doing it right now.
By B.
Yes. So essentially it is by supercoiling. It's just winding it up, making it more compact. There are more details with supercoiling and nucleosomes, how you make it more compact in the video I will put up a little later. I will put that up so you guys will have that to see. But yeah, that is pretty much what I have for now. Do you guys have any questions or concerns?
No.
Okay. And everyone did do the response, the form thing, right? That I sent.
Yes.
Okay, then you're free for the night.
Wait, I thought they said we had a quiz. Like, I'm not sure, but like people who took like people who had discussions where like we had like a quiz.
Okay, so the quizzes in my sections, I tend to like to keep them outside of class. So like it's like I'll keep the quiz online throughout the week. You can do it anytime during the week. I do it that way just because I rather go through more problems with you guys like these and talk about more about the concepts and make sure you guys do understand all of that. So that's just my perspective on this. I'd rather go through more problems with you guys than waste time during the discussion section itself with a quiz that might just tell you what you don't know. So
Thank you.
yeah.
I like her.
Thank you.
Not a problem.
Thanks.
Thank you.
Yep.
Bye, Lily.
Thank you.
Have a
Bye.
good week, guys.
You too. She's so pretty.
Thank you.